Okay, so can you just briefly describe the work that's currently going on in your lab at the moment?
Absolutely. So we are a traditional natural product chemistry lab. And so we are trying to find new small organic molecules from natural sources. And we're going to isolate those molecules and characterize those molecules. So we're going to figure out what they look like. And then we want to figure out if those molecules can be used in different therapeutic areas. At our core, we're really interested in finding new chemistry from mostly marine organisms. So we're focused on cyanobacteria. These are small ocean microbes. We look at sponges in the ocean, but we also look at some plants. So we have a really nice medicinal garden here, the Heber W. Youngkin Jr. Medicinal Garden. And we started an extract library of every plant in the garden. And we have looked at those extracts to see if they have any interesting chemicals. So we have one foot clearly put in the human health arena in terms of drug discovery, but we also have a little bit of another foot in looking at environmental toxicity. So say in Narragansett Bay here in Rhode Island, we have certain species of algae that will grow up and bloom, and they can produce toxins. Those toxins can get into the food system. So we're interested in understanding what species of algae are in the bay, what toxins are they making, and what concentrations, what levels are those toxins at, so that we can be sure the food supply is safe. So mostly drug discovery, but we like being a little bit in that environmental realm as well. We get to go out on lots of boats and things like that, so we're happy about that.
Very cool. Yeah, I was going to ask what types of applications your work had, both for the pharmaceutical industry and also just for the, society and.
Yeah, we think, really, the goal I think of most natural product chemists is to develop small molecule therapies for diseases. That is very difficult, it's challenging. You have a low percentage of these molecules actually go on to becoming drugs. But we're also trying to improve technologies that are used in drug discovery. So we try to improve NMR technologies, that's nuclear magnetic resonance, mass spectrometry. We do a lot of new mass spectrometry applications. So we're trying to enhance the tools that are used in drug discovery, along with trying to discover drugs ourselves. And also we're trying to learn more about the way cells work. So we're really trying to take a holistic approach. But again, at the end of the day, we are really interested in developing new small molecule drugs. So we are, I do have collaborations with pharmaceutical companies, and the goal really is to get something into animal studies and ultimately into clinical trials.
And what kinds of techniques or practices does your lab specifically use?
So the way my lab is set up is we basically have three different stations or three different areas. So the first is what we call the wet chemistry area. So that is where we are extracting specimens. And we're doing work in, say, the chemical hood, right? So we have extractions using organic solvents. We look at those extracts. We will use chromatography to separate those extracts into simpler fractions. So that's done on one side of the lab, basically preparing these extracts for testing. Then we will test them, usually in our core facility. So we use cell culture. So we will culture mammalian cells in the lab, and then we will test these substances on them to see if they can kill the cells. And if they kill the cells, then we may be interested in looking at them for potential anti-cancer compounds. So that's the biotesting area. So we have the extraction, the biotesting area, and then we have the analytical area. The analytical area is where we have our high performance liquid chromatography systems. So that is called HPLC. And that's where we're purifying out individual molecules. So that's a big part of what our lab does. We have three of those machines and we try to get them working as much as possible. When we have those purified compounds, then we go to our another core lab facility where we use mass spectrometry and nuclear magnetic resonance. And so this is going to let us know what this molecule looks like. Then we do actually have another area that's the molecular biology area. And here we're trying to make sure we can identify what microbes we're working with. So for cyanobacteria, we will amplify certain genes in the cyanobacteria that allow us to classify what we're working on. Or we might be interested in certain genes that make these molecules, the biosynthetic genes. And so we'll also use this area to identify those genes for further study. You're using lots of different techniques to get as much information as possible so that you can basically figure out what this looks like. You're basically putting together a puzzle. And so using all these pieces of information to work towards that common goal of saying, hey, when I draw this out on paper, what does it really look like?
And do you mind just speaking about some of the results that you've gotten or some of the contributions that your lab has made?
Yeah, absolutely. We've had really good luck so far in publishing new molecules. We have published at this point 25 new molecular structures. Most of them are from cyanobacteria. And we've been able to do some really interesting things with some new NMR experiments to figure out what these molecules look like, and some new mass spec experiments that allow you to basically visualize all the different chemicals that might be in an extract. So I think those are probably our biggest contributions are in terms of like structural chemistry. We have found a couple lead molecules. So we have a molecule that we're very interested in for its ability to reduce inflammation and its ability to bind to the opioid receptor, the mu opioid receptor. This is a target for a lot of pain relief. We have an opioid crisis currently, and some of those drugs are at a high risk for tolerance and dependence. And so there's a need for new drugs. So we're interested in that molecule. We have another one that is very good at, is very cytotoxic against breast cancer cells. So we're hoping that we can push that down the development pipeline as well. So a lot of projects going on, trying to always get the work out there to do really, interesting and innovative studies.
Very interesting. That's all really cool. Um, and in terms of the future, your future goals for your lab, where do you see your work going?
Yeah, what a great question. So I would say that we're really focused. We are focused on trying to develop these drugs further down that pipeline. So I would say in the next few years, I would love if artemisinin D is the molecule that we look at for this reduction of inflammation. If we can get that molecule or a similar molecule. So we have a project that we're going to synthesize different versions of the molecule to see if we can get better activity. If that can make it into animal studies, that would be a big goal of ours, is to get into the animal studies. The other goal is actually to be a little more in the chemical ecology area. We're very interested in a certain cyanobacterial species. It grows in the Gulf of Mexico, way out in the open ocean, like 300 miles offshore. And this organism is really important for cycling nitrogen. We're really interested in how that organism uses chemicals to grow and to cover so much of the ocean, because it's just so important for how the ocean ecosystem works. So that's kind of some of the chemical ecology we're interested in. So basically trying to get into that, into those animal studies and drug development, and into some of the more chemical ecology with some of these chemicals that we're finding.